co-directors of the first team of BrIAS. That was, he will tell us exactly the name because I don't remember. I will, please forgive me, but it was concerning agriculture. And last year, we had some collaboration in our first conference on smart agriculture. And this year we organized together yet another conference that was a huge success in this very same place concerning smart agriculture. And this year is also serving as a co-director again for the new program. So it's really with a big pleasure that I introduce Professor David Canella from ULB that will present us something.
Yes, thank you very much for the nice presentation. Well, first things first. Warning, that's a talk with a high content of cross-disciplinary information and so on. So you're going to be out of your comfort zone. So the comfort zone is a risk. Go take an extra coffee if you want. Yes, I'm going to bridge something on smart agriculture, biorefinery, all those concepts that, are kind of, up in the air, and try to bridge it. So to give you some focus on how one can help the other. To do that, I have to take it a very long way from, the car crisis that we have now in Europe. And then I have been told that I have 2 hours, right? How much I had? 45, 30, 40, 40, okay. I'll try to make a good use out of it. The last program that, the very long title that nobody remembers of the BrIAS, the first edition was Past, Present and Future on Food Production, Agriculture, Sustainability and Climate Changes. There you are. Maybe I have either lost or added one word, maybe. So, good. So let's say, I told you, I have to start it from a long distance. So you know these numbers. So these are the numbers of cars that we have in Europe. 250 million cars, more or less. Look at these other numbers here. I know that many of you are engineers, so I will flex my muscles. Ready for the battle. This is the max production that we achieved in the last 3 years per year of electrical vehicle. Around 2 million electrical vehicles per year in Europe. You having only Western, let's say, technology, not counting the Chinese, let's say, production system, or let's say that this car were also having some Chinese, let's say, or Asian, let's say, train production. So let's say some parts were done with Asian parts, but because European banned the batteries that comes from, let's say, Asian countries, let's say that these numbers, so the 2 million per year, are generally made with parts that come from the Western production. Do you think it's okay? I mean, look at the rate of production. This is the max we can do, the best we can do. 2 million per year, and we have to replace 256 million of cars. It will take more than 100 years, unless something changes. So, I mean, when people talk about the future of electric vehicle and so on, the future is already not born yet. I mean, like, the future is not even a reality. I mean, not even today we can talk about this as a reality. So we will have a big problem if you don't tackle this issue of sustainability in car in, let's say, in a serious way. This is not serious. I mean, going around and say that the future is electric vehicles when we can produce only 2 million per year, forget about it, okay? So something has to drastically change. I don't know if we have to change the way we produce batteries, the way we produce things, but let's say that that's not the future. So what about the other 250 million of cars that are still running with thermal engines? They are pretty much divided like this. So 70 millions of tons of fuel it is considered for gasoline car, and then under 80 millions of diesel car. Millions of tons. So if you go back to the original number, 250 millions of car, you might think that each car consumes sort of a ton of gas or fuel. Mm? But although we have done a lot of policies against diesel, you see diesel is still the one that takes the majority of the whole, let's say, fuel consumption in Europe. This is all European numbers. Obviously, because we have these heavy trucks that still runs on diesel. Road transportation for goods is the major one in Europe still. So let's say that although the private-owned cars are moving towards, or let's say phasing off diesel slowly, we still have a huge proportion of diesel consumption. Remember, these 17 million tons of gasoline will go back to that, this number. So now the numbers are there. So now I made up the numbers. So we have a lot of cars to replace, maybe not. We have a lot of thermal engines still, and we will not be able to replace all these thermal engines in due time to meet the requirement of CO2 reduction, for sure. And we are still consuming a lot of gasoline and fuel and diesel. When we look at where this energy comes from, you see that 60% of this energy, and we'll say like petroleum products, and so this 250 millions of, let's say, tons of fuel is actually here, is inside there. So that counts for the energy that comes from outside Europe. So we have the two first line, natural gas and total petroleum products, that they all come from outside Europe. It then makes up like 60% of the total energy consumed in Europe today comes from outside. Then you have the one that nobody wants to, let's say, still use, and we should actually ban it. That is the coal. That is the last one. Is solid phase fuel. So this is the last part of the, let's say, the energy, so the reminiscence from the past that we should actually phase out. And so the only two things that remain, let's say, interesting for European policies are the two in the middle, the renewable energy and nuclear energy. Those are the only two kind of technology, the source of energy, which are scalable in Europe, which we can make it in an independent way, and so make it, let's say, in a way that we could use this one to replace the 60% that come from outside. Now, what's the problem with these two kind of, let's say, source of energy? One is that the nuclear energy will provide you mostly electricity. And so for the electricity, you need like a lot of electrical devices to consume it. And I don't know if we are there yet. Again, the problem of the electricity that you have to consume it, and it's very hard to store it. So the stocking of the electricity is, again, a problem due for batteries. So now in the field of renewable energy, or let's say the new energy, let's say it, now they came out with this new way of thinking, this power to X, using electricity to convert this energy into a form of energy that is stable, that you can store it. Usually it's a liquid, an organic, carbon-made form of energy. So we'll come back to this one. So this slide was just to show you that the only two realistic solutions that we have in Europe are either nuclear or the renewable energy. When we speak about the renewable energy, we have all these 6 major sources. And all of them, so hydropower, solar, geothermal, wind energy, marine energy, they have in common that they are more or less, let's say, developed. We know wind energy and solar energy is much more advanced than marine energy. They all have in common one thing. They need a grid. They need a network. The electricity that is produced, or even the hot water that comes from the pipe of the geothermal, has to be distributed, so they need a network. Either if it is cable, or if it is like a grid, or let's say a pipe, still have to be produced. Again, we have this problem of stocking the energy. The only one, again, that can solve this problem, it is the bioenergy. The bioenergy that is one of the renewable energy, that is the only one that can provide you with a form of energy that is either in gas or in liquid. So when we speak about bioenergy, or let's say bio-e-fuels, we speak about converting biomass into either biomethane, that is now a very solid reality in Europe. We are actually doing very well in that. Or biodiesel, colza, palm oil, and so on, or bioethanol. And bioethanol comes from actually the sugar that comes from the plant. Right. So this is the old generation. When we speak about synthetic fuels, the classical fish and troughs, let's say, mechanism, designed during the First or Second World War, remember? taking carbon monoxide and hydrogenating this one to form, let's say, methanol and so on, but with a very high temperature. So this is, let's say, very inefficient at the level of energy. But it was good to create liquid fuels for the German troops when they had to fight the war. They were like under the embargo of, let's say, petroleum. So it's like one of those very emergency sources of energy. So forget about this, but don't confuse when people speak about synthetic fuels, they refer to this. So if they put the E in front, this is the new kind of E or synthetic fuel that is actually taking electricity instead of energy like temperature to do a similar process starting from CO2, but not the CO. So the CO2 that is atmospheric, hydrogenating it and taking the hydrogen because of the electrolysis of water using the electricity that is produced in a green way as a wind power and so on, and then produce again methane or methanol. So this is the future. So this is going to be the new E or the new synthetic fuels, and the E stays for the electricity that is being used for this process. What we have in the middle is this today. So today we are here in this, let's say, amount of energy that we can produce in a renewable way, bioenergy, that is liquid or gas, and this is a reality. I know that many people don't realize that we have a lot of technology already developed, but it's not so popular yet. So, but this is totally scalable. It's already like a demonstration plant. I will now guide you through all these, let's say, examples of these biorefineries that are, let's say, the base of what I was doing during my PhD and my early research career. So I'm really spinning out from this kind of research. And so I wanted to show you that when we talk about biorefinery and, let's say, lignocellulosic ethanol, or let's say second generation, third generation of biofuels, we talk about not anymore using something that human can consume as a food. So we are not talking anymore about potatoes or rice or sugarcane or let's say palm oil. We are talking about something that is only dedicated for the production of fuels and does not compete with food. That's super important. People still remember the infamous biofuels because they were produced, they still produced today, starting from food. But the new technology, the new generation of biofuels are all coming from, let's say, processes that do not touch any edible biomass. What we're talking about, mostly lignocellulose, so the part of the plant that we cannot digest as a human, because we don't have the enzymes that digest this particular part of the plant. So in French, we say la tige, in English, the stem, okay? So the woody part. The third generation that is mostly dedicated for the production of biodiesels, that is instead coming from algae. So these are all photobioreactors installation where you grow your algae that accumulates lipid, you open the algae cells and you take the lipid out, and these lipids are exactly the same kind of calorific power of diesel. And you can use this one to replace the diesel. And instead the ethanol and all the alcohols that we produce in the lignocellulosic degradation and then transformation into biofuels are there to replace the gasoline, okay? So what we are targeting when we do the processing of lignocellulose, we are targeting the cellulose inside the wood. So remember when you were in high school, so we all studied how the wood was made. Remember there was like the cell wall that is sort of, created by this very stiff and let's say polymer that is called cellulose. Some of you today still have some cotton in your t-shirt. I know that today is still like 80-90% polyesters, but the lucky of you that still have a cotton t-shirt might feel it. So that is the cotton, and that is made of cellulose. Then in a natural way, you have the hemicellulose that coat all around. And so this is what we call it hemicellulose or pectin marmalade sort of saying. And then you have the third component that is the lignin. So for decades, we studied the way to unlock the treasure of the sugar, so the cellulose, to penetrate there and then take this reservoir of cellulose and to expose it so to produce the glucose out of it. Glucose because at the end we will ferment this glucose in forms of ethanol. Likewise, the beer process. So it's pretty much brewing. The only difference is that in brewing, you start from the malt, that is very easy to obtain the sugar, the glucose. Here, we have to wean the millions of years that nature spent in protecting these cellulose, so to make the plant more resistant, so that we can have wood that stays there since thousands of years. In some of these archaeological wood is still untouched and not degraded by, let's say, bioaggressors. And so we can see that the whole nature, let's say, development have worked against us. So now we designed a fuel technology to get there and then to take this sugar. Why this so appealing? Because if we go at the numbers, a little bit of the numbers, I have to give it to you again. The whole production of this reservoir of sugar, of cellulose, lignocellulose, in Europe, considering only the cereals. I'm not talking about wood or other kind of, let's say, plants, just the waste of the agricultural system that we have in Europe. We are talking about 500 million tons of mass. 500, so this number here. We'll come back, don't worry. So that number there. So the 442 million tons, but we can approximate around 500 million tons. Only considering cereals, or let's say agricultural waste. Well, by doing the calculation and the considering the efficiency quite at the maximum, which is the state of the art of the today technology in the conversion of lignocellulose to final ethanol, we can obtain around 80 million tons of ethanol. That is equivalent to 60 million tons of gasoline. Only taking the full cereals waste of Europe, we can actually almost replace the entire gasoline consumption of Europe in form of ethanol. So you see now the appealing of this technology. It's there. That's why we consider the only scalable technology that we have in Europe if we want to replace tomorrow the gasoline instead and then using ethanol instead. So it's there. Now, how do we do it? It's a very simple process. Even master students now, they master it. So we offer a course here in ULB where we offer like 24 hours of a practical lab where they actually learn on how to do this process from A to Z. And so at the end, they go back home with a little bit of ethanol produced from either wheat straw or wood. So that's part of the course that we offer. And these are pictures from the students, actually. So this is like the wheat stem, like it is when you just harvest them. We actually do an hydrothermal pretreatment that is cooking. So cooking at like 100 degrees, no more, for a few minutes. Then we take the enzymes that are produced by fungi, which are our nanomachines. They are super fancy. They look like small dinosaurs with a cavity where they can extract like a single chain of cellulose and then chop the cellobiose glucose unit one after the other, walking through this, let's say, railway made of cellulose. And so these nanomachines that are developed by fungi are there. We usually walk in the forest to harvest new sources of enzymes, and then we simply produce this one using a little bit of fraction of the sugar that comes from the wheat straw. So again, the circularity theme is there. So we produce the enzyme by using some of the sugar, just like 5%, no more. And then we use this one to liquefy the wheat straw into a molasses that looks like that. The only difference between the picture on the left and the one on the right is that we added only enzyme. I mean, the water that you see on the right is the same amount of water that is on the left. It's only trapped by capillary forces, and so you cannot see it. But as soon as you digest the whole polymer, you actually liquefy it. Yeah, it looks like petroleum, but no, it's not. That is just a coincidence. What we want to do after that, it is a fermentation. So we simply add yeast, the one that we use for pizza at home, exactly the same. Then we obtain a transparent liquid that is the ethanol that is distilled out. So you have the bioethanol, which is not green, by the way, just transparent, let's say fuel, that is totally compatible with the new generation of engine. You simply put inside. Now, this one is sold as E85, ethanol 85% in few stations. In Europe, by law, so if you walk in any gas station, we don't have this E85, we only have, by law, a liter fraction in each of our gasoline. It's like 7, 8, 9%, maximum 10%. So if you now see on your gas station, there's a number, there's a letter E, and then a number after, that number tells you what is the percentage of ethanol inside your gasoline. So only few gas stations in Europe now offer this 85, okay? But we are getting there. I want to just drag your attention that this is the only chemical things that I will ask you to learn today. So it's the biochemical things that I will show you, how these enzymes work for degrading the cellulose, because it's a marvel, believe me. So these enzymes are just depolymerizing the whole cellulose by snipping this, let's say, let's call hair, or let's say chains of cellulose from the chain terminus. Let's say that they can recognize where the chains finish, so they enter, they penetrate, and then they start to chop after each other. And so they start to create this unit of cellobiose, they do unit of glucose, and then with this last enzyme, beta-glucosidases, it can cut down into glucose. That's not complete. You always need a gate opening, so an extra enzyme that creates the new chain termini, rupture into structure, so where the enzyme, the C-beach, the big enzyme can enter and destroy the whole cellulose. Those were the original cocktail of enzymes that have been developed until 2010, but they were struggling delivering, let's say, a profitable technology. Let's say that usually we stopped at 60-70% of degradation of the entire, let's say, lignocellulose. Not good for making money. Until 2010, when a pioneer group of scientists discovered a new enzyme, just one single enzyme, which changed everything. So that is one of the moments where you just find one single enzyme that completely pivots the entire field. So far we always talked about hydrolysis, so that this cellulose was cut by hydrolysis, adding water to cut the bonds between the glucoses. But with the discovery of the LPMO, that is an enzyme that is able to oxidize the cellulose, we actually started to gain much more efficiency, because this enzyme is able to cut in some regions of the cellulose where the other couldn't, and so synergize much more, and then produce, let's say, more chain entry for the other enzymes to destroy. So once we started to add this enzyme, we started to observe more beneficial, let's say, and better efficiency, that if we report here in sort of, by time and then efficiency, you can see that in usually 4, 5, 6 days maximum. So if you see the first black line with the square, that is the, let's say, the old generation of the cocktail, stopping around 60%. Instead, with the new generation containing this new enzyme, we can get up to 85, 90%. This is at suboptimal condition. At industrial condition, today we are talking about 95, 97% of efficiency of conversion of the original biomass into final product, that is the ethanol. That is like usually a jaw-dropping, let's say, reaction that I have from, let's say, chemical engineering, because usually in chemical engineering you have, like, when you speak about biomass, you're happy if you have 70, 60, 70% of efficiency, but 97% is super high. And so it became profitable. So we kind of surpassed the red line of profit. And so now they are actually running to create all this new demo plant. Don't get amazed by this picture that looks like, oh, fantastic, they are already like at the industrial level. These are just toys, okay? These are just small toys that, for example, this one has been built when I was doing my PhD in Denmark, in Copenhagen, nearby. Today is part of the Academy of Biomass Transformation that is like open there. So today it is a facility that students, new generation students, can go there to learn. It's not an industry at the level of scale and so on. So this is just like a toy. Very expensive toy, shall we say, but look at the numbers, what they can produce, 5,000 tons per year. we are way, way, way below the millions of tons needed. So let's say that they are not at real scale, so the real scale of this one should look like sort of 30, 40, 50 times bigger. And then if you have in mind what is a usual oil refinery as a size, I would say that this part here just occupy the parking lots for the personnel of a normal oil refinery. So it's very small. So, but these were instrumental to show that it works. And so now the good thing is that not only from cereals, so wheat straw, but you can see that many others started to use other kind of biomass. It's very interesting, the one in Estonia by Fimeron that is now using birch wood. And so also now the wood is entering the game. And so now we are unlocking another, let's say, box of treasure of lignocellulose that was not really tapped before. And then the one in Italy that struggled at the beginning, but now is catching up. Again, is taking marginal land, let's say marginal lignocellulose, the lignocellulose that is built in marginal land. In Italian we say canne, I don't know, like should be like giant reed in English. So those kind of, let's say, lignocellulose that comes from, let's say, marginal periphery of a cultivated land. So again, do not compete with first generation or let's say sugars and so on. Why we don't see them more? Of course, so the final price of this bioethanol, the E85, is below €1 actually, super attractive. The biodiesel the B95, it is a little bit higher than €1, 1.1, 1.2 usually. But we still don't see the many gas stations offering these products. I just zoom here on the region where we are living, the north of Europe. But I also saw a little bit in Germany, some of them, but they are not so popular. So there are maybe a few hundreds of these, let's say, gas stations. Reason why? CapEx cost, and then a very low oil price that is, quite of a big concurrence. Imagine that, this is the price today, but if you have to think of adding all these CapEx costs to produce this one, it has like a sort of inertia. So we will pay back, but the price that is now selling is too low. So the margin are very, very little. So nobody wants to put money from the pockets and then start to invest on this technology, having this little margin. So this is the major barrier for the biorefinery. And here comes the next phase. So trying to drag out of the fuel trap this technology. Because, in the meanwhile, we've been investigating all new source of technology. Believe me, like the way to unlock this lignocellulose with pretreatment, so the cooking, the enzymes technology, how to modify this biomass and so on. And then we just figured out that it was not only fuels possible to produce, but a full array of products that goes from biomass materials, advanced materials, drugs, and biochemicals, new chemicals, oligosaccharides, food replacement, prebiotics, and many, many, many more. So many that, at a certain point, there was a very good initiative that was already in 2005, if I'm not wrong, to regroup all these industries that works around the valorization of lignocellulose under a huge consortium that's called the bio-based industries, that today counts more than 300 or 400 established industries, not just spin-off, industries, okay, which are profitable and making product. Of course, then there is a lot of other spin-off. Each university today counts one small spin-off working on the valorization of biomass. That's the legacy we obtain from the journey of investigating how to make biofuels out of lignocellulose. And we have finally also a joint undertaking that is one of these, let's say, branch of the Horizon 2020, that is the Circular Biobase Europe, that is promoting and is offering a lot of, let's say, financement and calls and grants for, let's say, these thematics. So those are just small pictures for demonstrating you what we are doing today. So some of them comes from my lab and so on. So we learn how to make nanocrystals, nanofibers, so nano-sized, let's say, polymer out of this cellulose, controlling it with enzymes, of course. So you see, this is cellulose. Don't get Don't think that is a POMA. So this is just cellulose that is trapping water in a way that can become an hydrogel. So imagine that you can put your bioactive compound inside, that you can use this as a cream for delivering your drug to your skin. That's it. Transparent wood. Yes, we made it transparent without losing the, let's say, the polymer structure. So if you touch it, it's still, dense as wood, but transparent. And then we also learn how to use these new enzymes to crack in a specific way so that we can control actually the nano size, the size of the crystals that we are obtaining from the cellulose, again with enzymes. Instead of going to production of alcohol, we are actually now producing bioplastics, the PLA. Instead of making the alcoholic fermentation, just with a lactic acid fermentation, you can create the precursor for making the PLA. This is now produced in this way. And this is the last, let's say, revolution. So far I talk about only sugars, but don't forget that we also have lignin. They are the brown part of the wood that is today have been unlocked for producing all those new materials, advanced materials. So those are all these now common things that we can buy in any brico, which contain actually part of lignin injected, more than injected inside. Chemicals, as you can see, if you can manage to obtain the molecular, let's say, separation of these molecules, you can get up to 1,500 euro per ton. So it's quite, of a valuable, let's say, product. And finally, so this one brings me back to my last 10, 15 minutes, kind of. Yeah, 15 minutes of, let's say, applications that is in agricultural science. Because we just figure out that if we stop or we control the degradation of lignocellulose until a certain point, we do not go all the way to sugar, to the single monosaccharides, but we can stop until a certain point. That is the production of oligosaccharides. And so now the idea is to valorize these oligosaccharides in the way that can be like used as a prebiotics, or the latest, let's say, idea we got is to use it as a plant vaccine. So this is just like, take-home message. We can read it later on. I'm now excited to show you this plant vaccine application. I don't know if you heard about that, with these climate changes happening, we have more and more of these, let's say, wave of pathogens, microbial pathogen. I'm not talking about pests as an insect, but just fungal or microbial pathogen that attack the plants. So now we have today one of the most harsh to eradicate that is Botrytis cinerea. That is one of the most devastating pathogen that is causing more than 100 billion euro or dollars of worldwide, let's say, loss in equivalent in food. It can attack up to 500 plant species. So it's pretty much, there is no crop virtually that can be attacked by, that can't be attacked by Botrytis. So it is one of the most important to fight against with. Today, the state of the art says that we are using fungicide to tackle this problem. And so the idea is that, we should replace this fungicide with something else. These fungicides are usually very bad for our, let's say, body. Usually they even disrupt our hormone system, or they can even destroy completely the soil microbiome and render the soil pretty much almost, I won't say sterile, but very less biodiverse than it was before. And so the idea is to use something that is replacing it with, let's say, biologically derived molecule. The other biggest cons that we have with the fungicides is that they induce resistance. So year after year, you have less and less amount of little different kind of molecules that you can use to spread on the soil. But instead, the use of plant vaccine, it completely reverts the paradigm. We are not spraying against the fungi, we are spraying on the plant to teach the plant on how to protect the plant itself against the fungi. So it's just a way to awake, to stimulate the immunity, to tell the plant, hey, you are under attack, or you will be under attack soon. Raise your defenses, and then the pathogen will arrive in 1 or 2 days or 3 days. And so the plants are sort of, let's say, prepared for the fight. So this is the technology that we developed here at ULB. using these oligosaccharides that are, let's say, produced by the degradation of cellulose with these new enzymes that I just talked about. The first things we did is to test if it was working. And so indeed, yes, we saw that it was sort of protecting the plant up to a big extent, 70%. So let's say that the spread of the disease on the plant was reduced of 70% counter the plant that was not protected. So it has a quite of a promising effect. The good thing is that we learn all this by looking at the way the pathogen were attacking the plants. So we simply infected a plant one day, and then we noticed that by infecting this plant, this pathogen used the exact same enzymes that we used in the biorefinery for degrading cellulose. And then it took like, I want to be proud to say half an hour, and maybe 1 or 2 years to make the connection. That probably, let's say that, you know, the two things could have been very, very valuable, or could have been used for teaching the plant that could be relevant. So if you have, you know, the same enzyme that is attacking the cellulose when the cellulose is dead, but also when it's alive, probably is there where we should look at the source of this virulence factor from the point of view of the pathogen. So we started to study the biology of the pathogen. Again, cross-disciplinary. I dealt like with bioreactor design and so on, chemical engineering during my PhD, and then during the postdocs and the initial part of my career, we have been relearning all the biology since the fundamentals. So going back to the DNA, looking what is the most expressed gene during the pathogenic activity of these fungi, that was not easy. But we saw that, you know, this enzyme is fairly highly expressed during the pathogenic attack. So we isolated the most active. We used these enzymes to digest again the cellulose. We just grabbed all the oligosaccharides that were produced. And now we thought, okay, let's try to give these oligosaccharides to the plant to see if the plant recognizes as a signaling molecule for rise the immune defenses. So we tried, and then we succeeded in a good way, sort of say. So we observed that, you know, there was a lot of, let's say, again, protection. So the one before was on tomato, this is on Taliana. And so we have, again, a lot of protection going on. That is not enough. So to have a technology that works is not enough sometimes to, let's say, use it. Sometimes you have to really understand very well how it works. And you start to discover new features, which are, I have time, fantastic. Because now I think this is the most interesting things for the engineering coming on now. So one of the things that you learn by doing things for the first time in a field that you enter, like, you know, from yesterday, is that many things are already done. But then you have to really force yourself to search for the things that was not done. Then you spend more time in reading literature to see, okay, what is not done? And then you realize that it was like, you know, something that's very interesting, because you look in a way that the people of the field doesn't look the same things in the same way. You look as an engineering point of view, or let's say application point of view. And then we're like, okay, let's start to see if there is some molecules that we can use to crack the code. If this molecule can be used for, let's say, signaling the plant in a different or unusual way. And so we studied the way the fungus, let's say, attack the plant, and the way the signal is spread. So the first things that happen when the fungus arrive to the plant, it just trigger a very quick, let's say, signaling within the plant. And so this is a picture, not from my lab, of course, it's from Simon Gilroy from Wisconsin Medicine. He did this experiment just by putting like a caterpillar on a leaf. Then in a few seconds, like 20, 30 seconds, there was like a sort of messenger being produced in the plant that spread through all the plant. You see, in 20, 30 seconds, you have the signal that travels through, let's say, the cytoplasm of all the cells of the plant that being spread all around. So although the infection, sort of saying, the caterpillar started to injure the plant in one side, on the other side of the plant, the leaves were already ready to rise the immunity defenses. This changed everything. So imagine that if you can touch a part of a plant, and then the signal reverberate through the rest of the plant, that is very important. So now this is what we are doing. We are treating like only some of the few leaves of the plant, because we know that the entire plants can be then protected, or we are just simply treating the seeds, because we know that the mature plant that will come after the seeds being treated will carry on the defenses again. So this is a very important feature now. Few minutes later, so now we are away from this 20, 30 seconds, like few minutes later, you have an entire metabolomic reprogramming of the cells. The cells understand that there is something wrong with the surface, and then start to change completely the metabolism. It goes under inflammation, so this is one of the typical effects that have been always observed with the immunity. So also the plant get inflammation, and then at the end, after a few hours, 1 or 2 hours, let's say, the plants start to produce volatile hormones to tell to very distant part of the plants that they are under attack. Sometimes let's say the internal message travels, but then it needs a second message later on that arrives from outside to tell to the plant, okay, we are under attack, I confirm it, rise up your, let's say, defenses even stronger. But this one goes outside the plant, and is not only catched by the same plant, but also from neighboring plant. And now you start to understand that we can treat one plant and then influence the plant next to it. And this is not only observed with ethylene, that is this very small hormone, but also from other kind of gases, let's say, molecule. So this nitric oxide does the same. And the important thing is that, when you have multiple of these molecules arriving from different corner to a plant that was not attacked, it's just like rising more and more the defenses, and so it just will sort of create like a more complete shield for the next pathogen attack. So you have one plant that received the pathogen, it can, by bombarding of hormones, the neighboring plants, it can actually induce a full protection. The research on this regard is now wild. So we are now looking for all those molecules that can be, let's say, exchanged from plant to plant. So the very fast one are ethylene and nitric oxide, but now let's say this is a very small molecule, but now there are even specific metabolites that takes days to be produced. And so those one are usually associated with more, let's say, immune reprogramming at different stage. Of course, each plant has its own specificity. Each pathogen has its own, let's say, kind of aggression mode. So probably all this diversity on different kind of molecule that can be exchanged from one to another plant is not just like a universal language, it's actually different dialect that the plants speak each other. Okay, so you should see in this way. Then there is this very old theory, and that was already formulated in the 87 when they could not even understand this old part, because they had no molecular insights within this, let's say, theory or dynamics. So actually all those exchange of hormones is today, we are recognized today as the base of this push and pull theory, where you can pretty much grow your plant in an intercropped way, where you can put like some plants that are very good in repelling the pest, and others which are able to, let's say, attract them. Probably because they are exchanging the right, let's say, hormones that push the pest to fly away, and some other which are able to attract. So now, learning how to intercrop and how to, let's say, crack this code is super important. The last 2 or 3 important, let's say, basic information is that now we call this one cry for help, let's say, style. So we not only search on the aerial part, but also in the soil. So the same things happen in the soil, where the plants is actually producing an exudate that can recruit, let's say, beneficial bacteria. So this is, again, very interesting from the point of view of smart agriculture. So to enhance the property of the soil. And the last discovery, that was a couple of years ago, is that the plant emits sounds. Of course, yes, they emit sounds, but we are not able to hear it. They can travel up to 5 meters away. And so when the plants are injured or stressed, they actually start to emit sounds to to signals from each other. And so the neighboring plants can hear about it. And so the discovery has been done, thanks now to the engineering aspect of the entire field, because somebody have created like let's say a microphone device, or let's say an acoustic device, very precise, on the very low, I think it's yes, it's on the very low, let's say, range of sounds that human ear cannot hear. So you need to modify the microphone or the device, and also to create a model to recognize these sounds and to associate if it was like a sound produced for a drought stress or a pathogenic stress and so on. So now this model is now, let's say, just at this first example, is now going wild because each plant has its own kind of frequency and a kind of, let's say, sounds that can produce. And so this is now the new future trends in the next decade that will be developed. So not only hormones as a sense, so we not only need sensor for the hormones to understand what the plants are talking about, but also what they are really exchanging as a sounds. And so by being able to crack like this important, let's say, code, we'll be able to understand how the plants communicate among each other. Yeah. So just to go back to some few final slides, and to conclude the stories on how we discovered all these things from tomato actually. So is that we gave these sugars, and then we start to observe some reprogramming of the plant. We look at the DNA reprogramming, a lot of different, let's say, responses. The first thing that we observe is that the plants start to produce the fungicide. This is the biggest, let's say, sign of success, because we told the plants, hey, you're under attack. These are the part of your body that is, let's say, being liberated from the pathogen, kind of. And then the plant itself, even if there was no pathogen there, started to produce the fungicide. And so in case the pathogen could arrive, it would have found the whole surface of the cell covered by this fungicide. So very hostile, let's say, field to invade. We confirmed that by chance, and this is now not anymore a chance, but now is a reality, this whole technology works without any inflammation. So for the first time, we are observing a new signaling that does not induce the plants to go through inflammation. We are not yet understood why, so we're doing a lot of research on that. But we like this because, you know, without inflammation, the plants are not stopping the growth, so they keep growing and they go actually even faster because they're not dealing with something that they have to cure themselves. Yeah. Instead, they produce ethylene instead of the ROS signaling. And so we observe that even distal plants can be alerted. The plants are usually also protected for drought stress, so it's the same signal. So this is quite interesting for you to understand that, you know, different signals can give you different kind, the same signal can give you different kind of, you know, protection, not only by pathogen, but also from abiotic stresses. So these are the plants that have been treated that are much more resistant than the one untreated. They survived like 45 degrees in the oven, actually. Yes, and then just wanted to show you a few, let's say, picture of how they looks like the tomato. So we've done a huge campaign of experiment where we treated the plants with a several amount of these sugars at different frequencies and so on. And so we noticed that the plant started to grow actually and started to grow flowers and flower much earlier, like 2-3 weeks earlier. Since because they were not dealing with any inflammation, they could actually grow faster. Fruiting as well, so they started to advance the fruiting phase much earlier, 2-3 weeks. If you're not thinking about, you know, you are in a greenhouse and you can anticipate the production of your fruits by 2-3 weeks, so cutting down 20-30% of your time, so that's a lot of money that you can save. This is the way the plants look like. So the one on the left is always the one never treated, and so the more and more treatments, you can see that the plants go higher, that they mature the fruits earlier. So those sugars are all kind of, you know, model sugars that we have in our blend of sugars that we produce. The amount of fresh weight of harvest, so is also much higher. So we don't only protect plants, but we actually also stimulating the plants to produce more fruits. And so usually we have like up to 20, 30, sometimes even 50% more fruits by weight. And this is the way it looks like. So this is a classical plant that's been treated, and that one it was not treated, and harvested at the same time. So as you can see, you have many more mature fruits. And so as a number of, let's say, per gram of plants, we have up to 11, 12 grams per plant. That is like, for these particular plants, represent 20, 30% more fruits. So yeah, it is a sort of a payback. So you should not even convince the farmer to buy these things, because he's actually paying back itself. So the more money they make it, they can pay back the protectant molecule. Now it's over. So this was my last slide. As you can see, I try to bridge from going to a problem of substituting fuels with a technology. And this technology, by chance, also gave us new other kind of technology that now we are using for the application in agriculture. So we need a lot of cross-disciplinary, let's say, skills on how to apply these molecules. So the engineering aspect here is very much understudied, so it needs to be caught up. So these are some of the pictures of the people that have been working in this project. So the very early part of my group, and now the second part. I want to also thanks a lot Christian, that is down there. Has been my kind of senpai, or a senior colleague that have been driving me through this first 7 years here at ULB. So many thanks, and then I'm open for questions if you have.
We have a first question.
Thank you for this nice presentation. So you mentioned that these biofuels you are discussing are produced from non-food plants, but still there is a huge need of land to cultivate the birches or the whatever plants are used. And so isn't there a competition for land which could occur even though you don't use food plants?
No, because you are taking the plants residual of the food production. So you're simply living on top of the amount of plants already cultivated for your own primary product. That is food, or let's say timber for the building of a building, and so for this let's say sector. So you're only using the part that they do not use. So you're actually not increasing the demand of land for this. Of course, if then there is something profitable, then they will push for having more and more land cultivated for. But there is a lot of land available today used for the meat production. That is a response to the name of soja cultivation. That is only proteins produced for cattle, sort of saying. And so if there is a little bit of land to take, I would say the competition should go to that direction. So to reduce the meat producers. Yeah.
Other questions?
What's the catch on the vaccine plus bio booster? Because it sounds too good to be true. And I guess the question then would be, why hasn't the plant naturally evolved to do that on its own instead of being told to create that fungicide?
Because the fungi, they can avoid it. So actually the fungi, they can create countermeasures to shut down the immune responses of the plant. So if you just watch the two having like a fight, you will see that they have like numerous actually sort of signaling molecules that can one impede the other. And they are always at the minimum amount of concentration, just to overcome a little bit the defenses or the attack of the other. They are never at that big amount they can just cancel the fight and say, hey, now you raise the defenses. So this is what we can do as a human, control the amount of sugars that we can give it, like way more that is in a natural condition. Why? Because these are harmless for the environment. They are biocompatible. You can even eat. They are edible, these sugars. And so they are not causing any resistance to the fungi. That is the contrary of the pesticide. Where you have to be really careful how much you're giving and so on for not encountering these old problems. So, yes, it's too good to be true. It's true. But it actually works. So, and then we have the first generation of this is already actually on the market. It responds to the name of pectin and chitin and chitosan. So now they're already being sold a lot. I would say that virtually all the 99% of production of organic food is done by that, is sustained by these things. But there's only 2 products, with many more. So now this is the first that comes with cellulose, which is super abundant. The next will be the one based on hemicellulose, and then will be the one based on lignin, and so on. So the more of this biopolymer we can, let's say, unlock, the more, let's say, harvest we'll have to shoot back the pathogen.
Thank you.
Okay.
Thanks, David, for the amazing presentation. Now, from a chemical viewpoint, you talk about an efficiency of 97%. I guess that is the azeotrope between the ethanol and the water, no?
The what?
The azeotrope. I mean, at some point, the distillation of ethanol with respect to water,
Yeah.
you cannot continue distillating. That is why you have 97%. You can add, in this industry, they do add additive to improve that, because I know, for instance,
Yes, that's why it's
in
called
distilleries,
yes,
they are
yes,
moving
yes.
that azeotrope towards.
That's why it's not 100% ethanol, it's 85%, because you have to add an additive to reach to, let's say, so 15% is blended.
Perfect. And the second question is also about the efficiency, no? We're talking about 97%, but it's the transformation of biomass towards ethanol. But what about of the efficiency of each
Carbon.
joule produced with respect to each joule consumed? Because in this case, we have to cool down. We need a lot of enzymes, as
Yeah.
you said,
You're
fermentation.
If you ask this question, you're an engineer. So
Yeah.
you know that you cannot transform one joule to the same joule. I mean, you have to, otherwise you go against the physics law, the thermodynamic law, sorry. So of course you have to invest energy. And actually the price you pay for that, that's the price that costs. So you have to invest a little bit of energy to obtain more energy out. So, but this is what you are doing, is like you are transforming something that is solid in liquid. So that's what you are investing energy for. And then the majority of the loss in terms of efficiency is at the level of carbon, by the way. Because you start with a molecule that is a C6 carbon, you obtain the ethanol that is C2. So you obtain 2 molecules of that, so you have at the end, carbon, four carbons to be rescued. You start with 6. Actually, at the level of mass, you're losing sort of half of it. But let's say all these efficiencies calculated at the level of, let's say, amount of molecule that you can obtain. Yeah.
To be a bit polemic about that, if you have to pitch in front of investors for this process, and they tell you, what about biomass gasification? If you have biomass and you use it for ethanol, or you can use it to burn it because it's more efficient from an energy viewpoint, and you produce hydrogen. So what is the catch, as Marco said, to sell this technique or this process, let's say?
Likely, I don't have to deal with investors, so it's not my job. Our job is to provide, let's say, technology. My smartest reply to this question is always one, and so it's actually the dogma of the biorefinery. Never design a process that's only giving you one single, let's say, avenue of how to valorize your product into only one thing. Actually, biorefinery should be a collection of different kinds of processes that allows you to diversify your products and to actually adjust to the market requests. Now, because of the oil price going so much down, so there is no point of producing something that is competing with oil. And so could be that producing gases will be much more, let's say, profitable. But then you have to design your biorefinery to be able yourself to also go that way. So yes, the answer would be to have like the real biorefinery should have all of those, let's say, avenue possible, and so producing a range of different products: gas, liquid, biomaterial, biochemicals, not the single product. This will never fly. You're right. Yes.
Okay, so I have a last question, and then we will go for a coffee because we need it.
Yeah, yeah, no.
And the question is the following: in some of of these communication mechanisms between plants, is there any case in which plants that get a signal retransmit the same signal?
Huh, this is going to be the next paper. Not that I've seen it actually. So normally the plants, all those signals are only for the one I know, but maybe I can be somebody can know more regarding these topics. So usually they just prime the plant. So they just alert the plant to say, hey, you're about to be, let's say, under attack. But of course, if then there will be a pathogen that come and then attack the plant, now the plant will be ready to respond, and within the arsenal of responses, it will also again reproduce the same molecule that has been used for triggering, priming the defenses. That's for sure. But now the amplitude of the signal, is it the same or will be different because it has been primed and then attacked, responded, it won the battles, is the same or not? Is the same kind of combination of different kind of hormones, sounds, or let's say exudate, or is different because this is sort of second generation now, because you have the first one that has just been attacked and then send the molecule, but the second generation is the one that has been primed and then attacked. Who knows? This is a very good question, actually.
So with this, I'd like to thank again our speaker. I'd like also to close here the first part of these seminars. As per our tradition, we will take some coffee for around 15 minutes, and then we will come back here for our second talk of the day. So have a good coffee, and for those who are online, please have a coffee too and come back in 15 minutes. Okay, so welcome back, everybody. So it's really with pleasure that I introduce the second and last speaker of today, that is Luca Rossini. So Luca has been a junior fellows of BrIAS in the last 2 years. He is a postdoc here at ULB with a Marie Curie scholarship. And he has a quite interesting background that since I see in his preview of the slides, he will speak about it. I don't want to spoil. So I will let him the stage and let's see the show.
So thank you for the introduction, and actually thank you, David, for introducing many concepts that I'm going to recall in my presentation, because we have seen that plants should be protected, and so David showed us just one face of the coin, that is fungi, pathogenic fungi. So now I'm talking about insects, so about what insect science, called also entomology, but with a different approach. So the importance of models in plant protection against insect pests. And so, as David said before, so the same disclaimer of interdisciplinary approach will be also the same of my presentation, because this is basically my background. So I have a bachelor in physics, but then so I get passionate with the world of agronomy, and with my PhD I put everything together, because ecological modeling and applied entomology. And now, so since October 2023, I am a Marie Curie researcher at ULB in the Department of Control Engineering and System Analysis. So my Marie Curie project is called Pest Finder, and so our goal is just combining different disciplines to deal with the problem of controlling insect pest population in a farm. So basically applying all the techniques that are very well known in control engineering, but it's not just control engineering, as you will see, is insect science, is mathematics, is metrology, just with the final goal. So in this seminar you will see how over the years we arrived to formulate this project and what we are doing now. But I mean, as you said, as you saw in my first slide, I'm a hybrid person, and being hybrid is not always easy, because you have to interact with people with different backgrounds. So in my everyday life I interact with engineers, entomologists, applied mathematicians, metrologists, and everyone has his own language. So basically we are a kind of translator, so hybrid people act as a kind of translator. And we are also source of a lot of jokes, because, I mean, I heard many definitions of myself, because it's quite hard to explain who I am. Probably I don't know who I am. And, but just to give you an idea, according to the engineers, I am an insect guy who plays with numbers. But on the other way around, according to the entomologist, I am a physicist with 6 legs. So if you don't understand the jokes of 6 legs, just wait 10 minutes and you will know why. And so it's quite hard to understand what is an insect guy who plays with numbers or a physicist with 6 legs. So I thought that since one of the theme of BrIAS of this year is AI, why don't we ask to AI to explain what are those 2 definitions? And according to ChatGPT, this is the result. So it is something quite weird, but so according to AI, it is what I should be. But jokes apart, my main topic of research is rational pest control. And because now in smart agriculture, we want to we want to reduce the amount of inputs in cultivated fields, because every time we use an input, we are somehow modifying, so leading the sustainability of the environment, and we are also increasing the production cost. So basically, we should provide the inputs in cultivated fields only where and when they are really needed. And to do that, and in the particular case of insect pest, insect science alone is not enough. So we should combine different scientific fields together to look at this problem in a different way. And so, again, besides insect science, we need applied mathematics, because we can, if we have models to provide some forecast, we can know where and when we should provide the inputs in the cultivated fields. But we need also metrology, because we need to collect some data and some information from the field, because in this way we can combine everything together through control engineering and automation. And so in this seminar, I'm trying to explain how all those fields of research are connected in between each other. And I will start introducing the problem, so basically agronomy and insect science, and then I will move towards the second topic, that is applied mathematics, because I will explain which are the common techniques that we use to mathematically describe insect populations. We will move towards metrology, so I'll show you which are the complexities of collecting data in insect science and so in entomology, and then I will combine everything together with control engineering. So let's start from the first step. What is IPM, Integrated Pest Management? And the core question of this first part of the seminar is, so what is a model-based integrated pest management? So basically, why we need models? And if we have to describe what is, in an ideal world, integrated pest management, we can use a scheme like that. So we have a farm as a central point, because for a farmer, all the farming process is the core business. So a farm is like a company, and they should have some income. But from the farm, we can get some information through the monitoring. And monitoring can be, for instance, pest abundance, the infection level in the case of diseases, can be measuring the environmental parameters. So all information that we can get from the field. Those information can be somehow processed by models, and models can simulate different scenarios. Those different scenarios will be helpful to take decisions, and the decision is choosing the best control action, to reduce the level, the abundance of the pest into the field in a more sustainable way. So basically, we should start in IPM with the lowest impact for the environment control strategy, and then we should go, so we should increase the level if needed. The control action is going back to the field, but there are other two dashed lines in this scheme, because it's true that the information that we get from the field can be the input for the models, but models can somehow, and this is the role of control engineering, somehow suggest where and when we should collect the data. And this is something that, at least in insect science, is quite new. Again, if we include the control action, so the effect of the control action, into models, we can simulate different scenarios, and those different scenarios can provide more realistic, so we can provide a more faithful situation of the future situation of the field, such that the decision can be taken on more realistic, on something that is closer to the reality. But unfortunately, this is still a kind of ideal world, because the So if you look at the normal management of farms, this is the real scheme. So basically you have an approximate monitoring, so a few information that comes from the field. And based on those few information, the core model is not an equation, but is the experience of farmers, because they are so experienced in sometimes that they can process the information and they can find the best control strategy. But I mean, it is something that is still, so for the level that we want to reach now with precision agriculture is something that is not anymore sufficient. And this is the reason why we should improve the scheme, developing models and new techniques to move towards what we call decision support systems. And so now, this is our main goal. And smart agriculture from the pest control point of view is solving this problem. And this is the reason why we need all the different, so the interaction of all the different fields of research that we have seen at the beginning. But now, so we are moving towards the second part of the presentation, that is mathematical models. So from insect science, we are moving towards applied mathematics. And the core problem here is to formulate reliable models with a biological meaning. So biological meaning is the most important part, because we don't want just a mathematical game that more or less reproduce the trend of the data that we can collect from the field, but we want that the biology is suggesting us the assumption to build the models. So, because, I mean, we don't want to lose the connection with something that we can observe and measure. And since the biology is the main component of the formulation of this problem, let's start from scratch, introducing what is an insect. And if we want to describe in a very simple way what is an insect, we can say that is an animal with 6 legs. So just now you understand the joke that I made at the beginning. But from the modeling point of view, there are few features that we can account, and the most important one is that insects are ectotherms. It means that the duration of the life cycle depends on the external environment, mainly depends on temperature. So at low or high temperature, we will have longer life cycles. And then at the optimal temperature, so for all the species, we can always identify an optimal temperature where the life cycle is the shortest one. And this is one of the main features that we should account if we want to build a reliable model. And then there is another feature that is really important, that the life cycle of insects is divided in stages. Those stages are clearly identifiable. So if you ask to an entomologist, can you explain how many stages compose the life cycle of this species, you will get an exact answer, because all the stages are morphologically identified. Then we have more or less 1.5 million of species, and we will have very different and complex, less complex or more complex life cycles. And not all the species have the division in males and females, because there are some cases where there is a phenomenon called parthenogenesis, where females are just reproducing females for some species. So the sex division is something that is not always happen. And now, this is just the first point, but this is a hint for all the people involved in image recognition or computer vision. If you want to recognize an insect, you have to keep in mind that there are, in all the species that we have different sizes, so from a fraction of a millimeter up to 20 centimeters. and different shapes. But those different shapes are, so we can have different shapes between the different species, but also different shapes in between the different stages of the same species. So an egg, the shape of an egg is different from the shape of an adult. So you should keep in mind the stage that you are targeting and that you want to recognize. And insects are everywhere. So if you think that it's just a problem of natural environments or of agricultural fields, this is not the case. So insects are, of course, in natural environment, of course, in cultivated fields, but they are everywhere because they can infest greenhouses, they can infest warehouses, we can infest our houses because there is a branch of entomology that is just facing problems in urban environments. Think about all the vector-borne diseases. It's like malaria, Zika, this kind of viruses. They are all vector-borne diseases. So it's a mosquito, the vector. So basically, again, is insect science. But now, more or less, we know what is an insect. Let's see how can we obtain more or less a good model to describe mathematically an insect population. So this is something that I reached more or less towards the end of my PhD. It was during my first year of postdoc. Because analyzing the literature, what you see, the old literature, what you see is always a single model to describe a single species. But actually, if you look at the difference in between, and the analogies between the different life cycles, so from the biological point of view, and the equations, so the mathematical point of view, there are a lot of common points. It means that we can, so generally speaking, we can put all the mechanisms that we can find all together in a single scheme. But when we know, for instance, how many stages compose the life cycle, or we know the mechanisms of reproduction, or for instance, if we have which is the sex division, so if we have males and females, or if we don't have the male, we can obtain the particularized model. So this, which is the philosophy behind this scheme? So each compartment represents a single life stage. So a life stage that if you ask to an entomologist, can you define what is an egg? So basically, from the mathematical point of view, is this compartment. And so this compartment is described by an equation, and the solution of this equation is the number of individuals over time. This number of individuals over time and in all the stages basically is regulated by some transition rates. Those transition rates are development rate, if we are meaning the transition from the stage to the next one, can be a mortality rate if the individuals are living the life cycle, can be a reproduction rate if we have a production of new individuals coming into the first one. And so we can also have the sex ratio, because so the sex ratio is the ratio between males and females, and is not always 50-50 in some populations. The extreme case of parthenogenesis, the sex ratio is 1. It means that this term is disappearing. So this chain of compartments is a single line up to the last pre-imaginal stage, so the last young stage, and the division happens only when we have the adult. Why? Because it makes sense dividing the population in the adult just because it's the only stage that is devoted to reproduction. So it doesn't make sense to have twice the number of equations for something that we cannot observe. Then, in the female stage, we can have different mechanisms. Again, different species, different mechanisms. We can have, for instance, a division in between non-mated females and mated females. It means that the beta 2 transition rate is set to 0. So we are canceling out this arrow here. Or we can have different fertility rates based on the aging of the females. So it's what we call gonotrophic cycles that is typical of some species of mosquito. So as you can see here, we include all the possible mechanisms, but if you don't ask to an entomologist to define the life cycle and the main mechanisms, you cannot apply this scheme to a particular case. And this is what we mean that we need biological assumptions. From a mathematical point of view, this scheme is just represented by a system of ordinary differential equations, and the key role is played by the transition rates, because those functions, generally speaking, so the parameters of those functions can be estimated through laboratory experiments. So those experiments are quite complex, but so we can obtain some parameters, and they connect the mechanisms that we have in the population with the external environment. So the Trojan horse for the environmental parameters is inside the transition rates. This system can be non-linear. It's not necessarily linear, because if you have some dependencies, for instance, on the abundance, so the population abundance of some states, you can use the transition rates to include this dependency. So this was just the first step that we carried out, so we were quite surprised, because at the beginning we applied this model in different case of studies, and we were surprised to see that at least we were able to reproduce the trend of the data, and there is no correction from the field data. It's just an open loop simulation. But again, so precision agriculture is providing inputs where and when. So from an ideal point of view, we could just answer to the question when we should apply the treatments. We are not answering to the second question, that is where. And a first attempt of spatialization so just comes from analyzing the mathematical structure and the stability of the system. And as a result, you can obtain a risk index. This risk index is parameterized, contains all the transition rates, and connects, again, the environment, so the environment and the parameters that identify the species. But this is something that still is not practical, because it's not considering one of the most important parts that we need. So basically, we need to know how insects move in between the different areas of our field. This is the reason why, again, we went back to entomology, to insect science, and we started to analyze which are the most common approach that are used from insects to move. So, as you can imagine, there are a lot of different mechanisms, and so whatever is the species, you can always identify which is the mobile stage. So the stage that can cover long distances. We can have, for instance, passive movement. So there are stages, some stages that is like a powder is carried by the wind or by water. In other cases, you might have an active flight, so an adult with wings. But anyhow, you can always identify which is the stage that is forced to stay in the plant. It's like an egg, so an egg cannot move. While an adult with wings can, so is devoted to spread the outbreak in other places. And I mean, if you identify those features, the game to spatialize the model is to use what we call a meta-population approach. So you can divide your field. Can be a single field, can be a bigger area, but so the concept is exactly the same. And in a series of, in a grid, and you obtain some subparcels. So each subparcel can be described by the system of equations that we saw in the previous slides. But now we have some migration rates that appear only in the equation that is identifying the mobile stage. So two parcels can exchange individuals if and only if they share one boundary. This is the strategy that we use to spatialize the model. And this was a good step in the theory, but up to now we are still looking for some good data for validating the model. And as you will see, collecting the data is a big problem in our field. But now, we could answer to where and when, but still we have a problem, because so once we were just looking at some data collected in a climatic chamber, when everything is controlled. You know how many eggs you had at the beginning, you know exactly which is the temperature, the diet, all the things that you cannot control in a natural environment. And this is the situation that you that you see from the data set. Basically, you can track the population over time in all the stages, and you know exactly which is the initial condition. So, but as you can see here, the peaks of the population in the different stages, they are not overlapped, but there is a time shift. So this time shift is something that is recalled in the biology. So if the development is something that is related to a series of biochemical reactions, even if the environmental conditions are the optimal ones, we cannot go, so we cannot decrease this time that we have to wait. So this scenario was quite mind-blowing at the beginning, because we said, okay, we want to test the model, because usually in a field, in an open field, you just track a single stage, but you don't know anything about the other stages. In a climatic chamber, you can track all the stages. So this is the perfect bench test to validate a model. But if you look at the simulations here, so this is not the simulation of this data set, it's just a plot to show you the behavior of the simulation. All the peaks of the different stages are overlapped. So we don't have this time shift. So again, we made a little step back, and we started from the beginning. And we said, what is missing? Is missing an important concept, that is the minimum development time. So if we place 100 eggs in a climatic chamber, and those eggs are laid in the same moment, at the same moment, so the minimum development time is the time that we have to wait to observe at least one egg that is hatching, or a transition to the next stage for the other life stages. So from the mathematical point of view, this led us to revise a bit the system, and from a system of ordinary differential equations, we went to a system of delay differential equations. Let's say that this is more or less the philosophy behind our modeling approach. And as you can see here, so this is a simulation just to show you the behavior of the model. Now you have the time shift. So it means that the concept of minimum development time is something that is really important if we want to improve simulations. But now there is a common belief in many, so talking about with entomologists, there is the common belief that if we have a model and this model is validated, this model is ready to be used to be included in a decision support system. So basically, if we can describe a series of experimental data, we can use the model for predictions. And now we have this question. Are these models ready for predictions? The answer can be yes, but if and only if you know which are your initial conditions in the field. So basically, you should estimate in a clear way when the outbreak, so when is the time zero of the outbreak, and the initial abundance of the population. This is it is something that, trust me, you cannot know easily. So basically, we are moving towards control engineering. But before doing that, what can be the solution? Probably we need a feedback from the field. So now we are moving, before jumping towards control engineering, we need an intermediate step through metrology. Because if you think that the data collection process in insect science is something that is easy, the answer is no. And now let's try to answer to this question. Let's try to understand what is a measurement in insect science, and then we are trying to find a solution to combine the models and the few measurements that we can get. So, what is a measurement in insect science? And there are different techniques, and I will show you the pros and cons of the different techniques. I want to introduce the first one that actually is one of the most common, that is the visual inspection. Visual inspection basically is defining a protocol and a certain number of plants that you want to inspect, and a stage that you want to target. And just to give you an example, so let's consider a vineyard. And this vineyard is composed of 2,000 plants per hectare. And we want to track the population of one of the main pests of vineyards, at least in some areas of Europe, that is Lobesia botrana. And you can see why it's an important pest. So basically, it's causing, besides the direct damage, is also opening the door to a lot of molds. So basically, what we call secondary host in this case. So your protocol can be inspecting 5 bunch of grape per plants, and if you are super expert, you can, each measurement is just taking 30 seconds. But this is just for one single point, and if I'm not bad in math, you need 80 hours per week, because you don't want just a single point, you want a time series. So you should repeat this operation for all the growing season, that usually is 6 months, 5 months, it depends on the crop. But if you want to track the population all the year, you should repeat this operation all the year. So it is something that is extremely time consuming. And this is still a kind of lucky case, because the height of a grape plant is something like that. So something that you can easily reach. But imagine that you want to track the population of this guy, Tomaiella parvicornis. So this guy is 2 millimeters long, and is basically a huge pest of stone pines. So it's an alien species for stone pines, and it's a huge problem in Rome. So I was involved in some research, and I can say that the damage is something that is really, really huge. But the living environment is the canopy of a stone pine. It means that if you want to take some samples, you need to use a crane. So basically you have to rent a crane, you need someone with a special permission that is going up, is taking samples, you have to go to the lab, count the specimen on the different samples, and you repeat this operation on different plants and all the weeks. Another technique is quite curious, that is called frappage, or branch shaking. So I used this video from a colleague, Nicolò Di Sora, and you will see this guy many times in this in this presentation. And so it consists of having a white sheet on the ground, and you just shake the branch of the plant, and you are going to count how many individuals are falling on the sheet. So you understand that you should repeat this operation for a sufficiently high number of plants in your field to have a good statistics. But again, it's just for one single point. So you have to go there every week and you have to repeat the operation. But so likely enough, we have some good friends that are traps. So traps is a kind of way to automatize the data collection process in entomology. But again, the process is very easy to understand. So there is a lure, and this lure usually is reproducing the smell of food or the sexual attraction. So it basically is reproducing something, some biological aspect of the target species. So with the lure, we are just attracting the individuals, and then there is a catching part. So usually it's a sticky band like this, or is a bottle. And so on the sticky band or inside the bottle, we can count how many individuals that we had, so that we caught. And but every time you have to inspect one trap, you have to do this game. So basically, you go there, and again we can see Nicolò doing the job, counting how many individuals are on the sticky band, taking notes, and doing this operation for all the traps that we have deployed in a field. So this is something that automatizes the process, but still there is one common problem to all the techniques that we saw. So we don't know which is the proportion of the individuals that we are counting and the actual population into the field. So at the point that recently we had a bit of brainstorming about that, and so we raised a question. So what is measuring a trap? And this is actually an open question, and is an open door for all the metrologists. So please help us, because it is something that we still don't understand. If you think that just counting insects is something easy, you're wrong. Because so the identification of the species is something that is extremely difficult, and there is a branch of insect science that is called taxonomy that is just defining all the morphological and genetic traits to classify the different species. And so usually so insects are classified based on the morphological traits. It's not just looking at the color or a general shape, but it's something that can be, so it's really analyzing the shape of a leg, the shape of the wings, the shape of the antennae. So, and in this way you can define which is the species. Just to conclude the part related to measurements, so in all the cases that we have seen, we are just counting directly the individuals. But there are some cases where this is not possible, because we can only see the damage. So this is a case of it's not an insect, it's a mite, Phytoptus avellane, but it's a mite pest of hazelnut plantation. So what you can see is the damage that is called pseudo-gall or big bud. So basically so this guy is going to infest the healthy buds, and the plant as a response is producing the big buds, so the pseudo-gall. But actually, if you dissect one of those galls, you can see that you have a colon inside. And this is really something small, something really small. And if you want to spend some time at the stereo microscope to count the individuals, this is the scenario that you see. So basically all those small points here are the mites. Again, here the only way is just counting the galls and trying to estimate the infestation level. So it's true that measurements in entomology is something that really needs help. But those information can be somehow used and connected with measurements, so with models. And the answer is yes. And so we are moving towards control engineering, because the first time I interacted with Emanuele, I was always trying to look for a solution to estimate the initial conditions, to switch from the description to the prediction. And so the answer was, yes, but it's our job. Okay, so we started the interaction. And so basically, the idea is to revise the model and to build a model that is reproducing the measurement process. And the first attempt that we did to solve the problem of the initial conditions was applying one of the most common techniques in control engineering, that is the Kalman filter. And basically, it's a two-step algorithm. I know that there are control engineers here, so please don't blame me. And but so just to explain in simple words, is starting from the assumption that a model is something wrong by definition, because it's just a schematic description of a natural phenomenon. On the other way around, measurements are affected by an uncertainty, but the Kalman filter is trying to combine those two information to obtain an improved simulation. And so we applied this scheme, so this framework, to the case of Drosophila suzuki. That is exactly the case that we saw at the beginning in the first validation of the ODE model. And so we compared two scenarios. The first scenario is so randomly generating some initial condition and solving the model. And as you can see, the dependency on the initial condition is quite hard. So it is quite high. So there is a huge variability. And we cannot, of course, we cannot rely on a simulation like that to take decisions. But if we include the feedback from the data, we are we are reducing the dependency on the initial conditions. And we are improving somehow the trend of the simulation. So the trend of the simulation is closer to the field data. So this was our first attempt of applying control engineering to insect science. And but then this opened a lot of problems that are really interesting, because now we are moving towards the last part of the presentation, that is more or less what we are doing now. So putting everything together. So we have models, we have some measurements, and now we know that we can combine everything together through control engineering. And the problem one of the problems that we are facing now is the optimal sensing problem. Because one of the first questions that I always heard when in insect science, so entomologists are starting a monitoring survey, is how many traps I should use and where I should place them, because this is one of the most recurring questions. So what we are trying to do now is to use the properties of the Kalman filter to solve what is called an optimal sensing problem. So we want to define, given a number of traps that we can afford or that we can manage. So should we leave them in the same place, or can we move them to improve the information that we get from the field? So basically, this is what we are trying to do. So it's a model-driven data collection. So asking to the model where we should put the trap at the next step. Then another. So this is the last slide. Another problem that we are facing is it's a farming problem. So there is a common belief that usually the pest population should be the lowest one into the field, but actually this is not the truth, because the core, so the main goal of a farm is to maximize the profit. And I mean, if we keep the population low into the field and we use control, so treatments, so this is just increasing the production cost, and sometimes it's not worthy, so related to the yield that we can gain. So basically, we should build a model that is taking into account the plant as well, so the yield, and so the model now is going to be a kind of predator-prey model, where the insect pest is the predator, our fruits are the prey. So based on that model, we can formulate an optimization problem, and we may wonder when is the best time. So given a number of treatments that we can afford, and sometimes it's something that is fixed by the law, so we cannot go beyond a certain number of treatments for a given pest in a given crop. So the first question is, we need 3 treatments, if 3 treatments are allowed. The second question is, when is the best timing? And this is something that we can, so it's a question that we can answer through an optimization problem. So this is another ongoing work. So just to conclude, as you can see, so as you saw in this seminar, insect science cannot be left alone, and it's just combining and looking at the problem from different point of view that we can have very good solution to the problems. And with this I have concluded, and I open for questions, and thank you for your attention.
So, thank you, Luca. Are there questions?
Thanks for the presentation, Luca. So it's again from a profane point of view and a neophyte in the field. So by approaching, let's say, pest control and pest monitoring, there's always sort of a question that arises. So it's clear from what your presentation that it's very hard to do pest monitoring with the, let's say, with the visual inspections and so on. It's quite, an abnormal amount of work. But having some devices that, check for you, you will go for the single pest monitoring or the disease that the pest causes. So do you also integrate this kind of, let's say, in your model, so this kind of, inspection, so to see if the pest is also virulent state? Because we always, associate the presence of the pest like, okay, the presence is enough to tell you that it's causing the disease, but you should also check or not the presence of the disease.
Okay. So you mean with pests that are carrying diseases, or
no,
is just the damage?
Damages.
Okay. No, because so with the pest vectors, yeah, this is this is something that is extremely complicated. But yeah, so this is one of the main problem actually, because you should relate. So in classical integrated pest management, so there is the concept of economic threshold. So it's a certain level of the population that I should not overcome, because after that threshold, I will start to lose money. But this is something that is quite empirical. So the idea, so what you say is true, yeah, because the pest so a high pest abundance, we always do the assumption that if the population is high, the risk is high, so basically we will have damages. But yeah, so we should look at the damage as well. So, but the damage is something that is indirect, because it's really hard to correlate the pest abundance that you have 3 problems. The first one is the connection in between what you observe, what you count, and the actual population into the field. The second problem is connecting the damage with the actual population. The third problem is connecting everything together. So for some species, let's say that the damage, so they consider the threshold as the number of fruit per plant damaged. That is something that is related not on the pest abundance, on the population level, but is based on something that you can observe. But yeah, so in my opinion, that should be interconnected. But the problem is, how can we connect them? So this is the big open question.
There was a question there.
Thank you. My question is, considering the fact that we can also have multiple pests that can be of infestation in a field, and you also have multiple environmental factors, you have different growth stages of the pest. Is it possible to get a model that can cater for all these challenges that plants may face through pest infestation?
Okay, so in the case where you have multiple pests that are infesting the same plant.
Yes.
Is that the question? Okay.
Yes.
Yeah, of course you can. Because the general approach that we tried, so that we formulated, is something that can be, so that can describe the life cycle. Now, so besides the optimization problems and optimal sensing problems, what we are trying to improve is the different trophic levels. So connecting the different trophic levels, because one case can be when you have this is a kind of double predator-prey model, because you have two predators that are, and the prey is the same, that is the plant. But in other cases, you might have three trophic level, so plant, pest, and natural enemy. And this is something that is extremely interesting, because when I talk about control strategies, I don't mean just only agrochemicals.
Okay.
It can be whatever control strategy. So entomopathogenic fungi, predators, parasitoids, and of course connecting multi-trophic levels is something that is really interesting. And yeah, we can do it. We can do it. Yeah. With the same approach. Yeah.
There is another question.
Thank you for your talk, Luca. You showed us a patchwork of plots with various levels of populations, and you made the assumption that only the plots which had a boundary with another plot would have the pest communicating.
Yeah.
But of course, we have also situations where the situation is much more blurred because the pests are extremely mobile. In this case, we can have a patchwork of damage which does not translate in the same patchwork of pest abundance. And how is it possible for, what you call that, control engineering to solve this type of issue?
Actually, it's a question for you, probably. No. I will try to give my point of view. Yeah, so it's true that, as we said at the beginning, so when we were talking about the transition rates, so in between the different stages, so the development, the mortality, the philosophy behind is that this is a kind of Trojan horse that to connect the biology and the population dynamics that we can observe. So the transition rates in our spatialized models more or less is the same. But while the biological knowledge on the thermal biology, so basically how the temperature or other environmental factors affect the development and the mortality and the other transition rates is quite well known, at least for some species of agricultural interest. So the spatial patterns is something that depends on measurements first. So basically it's a question mark. The second fact is that we don't know exactly all the mechanisms of migration. So it is something that should be explored a bit more, because then the challenge is estimating the migration rates. This is the main point.
Maybe if I can complement, as a matter of fact, so the model is with some migration that is possible and some other that is not. Of course, if I know that a species can at most do 200 meters per day because they walk, you can have a graph of how these things evolve that depends on that. And then you have a lot of surprises. For instance, lately we received one of the very few specialized data that we have ever seen. And there were some patchwork that we could not understand until we spoke with the person that collected the data. And she told us, yes, this is in a greenhouse. And we discovered that there were these mites that were walking on the structure of the greenhouse. So absolutely, I mean, the mechanisms there are much more complex than what we can measure in a lab.
Yes, I mean, there is something that, while for the description of the life cycle, we can be more or less general enough. So, and then we can go to the specific case. Here, I think we should face case by case, because then you have also the cases. So, depends on the scale. So, a field scale, a greenhouse scale, and this is a first context. Then you can have some long migrations where you have some individuals that are carried by winds or current, and it's quite common for a lot of species. So your initial outbreak depends on the meteorological factor, but at the macro scale. So yeah, I would say that it's quite unexplored and interesting to explore.
But there is one thing to be said, that, for instance, for geographical movements, the thermal aspects can be interesting to understand when there are bridges between geographical areas that usually don't communicate. So if I have a mountain range that I have to pass through, maybe it's a summer that is particularly hot, the summer in which something goes on the other side. So there are these aspects that anyway come back from the biology.
So, I mean, it's millions of years of coevolution. So, yeah.
Any other question? I might know. I'm joking.
Oh, you can.
Okay, so I would thank again Luca for his talk.
Thank you.
And with this, I close here the formal part of our Thursdays at BrIAS. As usual, this is only the formal part, in the sense that you are all invited to continue get another coffee, because probably you will need it again, and to chat with the researcher, to meet our fellows, to speak with others, and to try to create connection and good, nice ideas. With this, so I also thank all the people that have followed us remotely, and have a good afternoon.